Hello everyone, my name is Shubham. I have aimed for Junior Research Fellowships CSIR June 2022, and in this video I'm going to talk about a topic from our Unit 12, Applied Biology. Let's get started. So today we are going to discuss about gene therapy. So as you hear the term gene therapy, two things come in your mind: DNA and doctors. Well, here in this special case, the credit which should have been gone to the researchers more often is due. But anyways, we have to have a basic understanding of this particular approach of how we use molecular biology to kind of, or the biotechnical application of molecular biology to kind of get help in case of certain rare genes which get defected and cause malady in us. So basically you have to have the idea of there are basic two types of gene therapy or genetic engineering as per, which is gene augmentation therapy or gene inhibition therapy. Okay, so in as the name suggests, gene augmentation, you are actually knocking in certain kind of you know external influence that is leading to get the treatment for the disease by making it not functional, non-functional. So you're basically using knock-in mechanism here. You're using the knock-in mechanism in gene augmentation therapy. Whereas in the gene inhibition therapy, you are just doing the opposite. You're basically treating the disease by making sure that you apply certain gain of function mechanism and knock out the non-functional gene out of the system. It's more like a Kree-Loxp system that you have all might have heard about, you know. And yeah, so basically for doing this kind of procedures like either gene augmentation or gene inhibition, you have to have basic approaches such as you can use a germline approach. Either you can use a germline approach, right in the gametic cell itself you can incorporate your influence or your effect through the techniques, so that either by the knock-in method or knockout you just get the nivaran from your problems. And the somatic cell approach, where you incorporate this particular amends into the somatic cells of the organism. Okay, so as we can see that if we incorporate this kind of amend or improvement in the germline cells, it will be incorporated in the future generations as well. But in the somatic cell approach, it will be only for that particular child or patient which is having the, or who or she or he having the problem. Now uh, there are certain vectors which you use here. Are, again, once again, biotechnical applications, vectors are the most important thing. So here we have viral vectors which are mostly used. These are different kind of viral vectors that can be used, such as retrovirus. You can use adenovirus. You can, but here this, in the case of adenovirus, there is a requirement of re-administration. You have to re-administer this adenovirus after a certain period of time. You can use adeno-associated uh, system, which is specially found on the chromosome 19 in humans. And herpes simplex virus is also used. This is used mostly, this is a double-stranded DNA vector, and this is mostly used for neurons delivery. Physical, now when we use non-viral methods, there are some certain methods, like physical method is there. There is a chemical method, liposome mediated therapy are there. So you can use these kind of things as well. Now uh, one example of this somatic, there are two types of somatic cell therapy that you can use. Either you can use it in vivo or ex vivo. So if you're going for in vivo, like for example in the case of you CFTR, cystic fibrosis membrane regulators. When they get defected, you use an adenovector, and that leads to the help of the cell, so that more chloride is not deposited, and the balance is maintained, the homeostasis is maintained. One more thing, then the ex vivo part, there is a special case of a child in India that is known by the name of Shanti Tishilva. We have used the ex vivo technique here with the help of adenovirus. This was the case. The child with SCID, he had severe combined immunodeficiency, and so we isolated the cells, lymphocytes, and we incorporated a vector adenosine deaminase genes, retrovirus, incorporated it in this vector, and then the lymphocytes with corrected adenosine deaminase genes were cultured, and they were cultured to verify the expression, and once it was verified, they were injected back into the patient, and this led to the synthesis of adenosine deaminase correction of severe combined immunodeficiency disease. So we need to have proper understanding of what is the constructs that we use in these cases, the kind of constructs that we have to use. See, in a viral genome particularly, there is the 5' end, there's a 3' end, and there are the primer regions and the origins of whatever that might be the replication recognizing part, where the protein assembly happens, where the RNA assembly happens. But there are certain RT precursors which lead to the reverse transcription, and that leads to the hijacking of the host machinery. The capsid precursors which lead to the development of the coat. We need to get rid of these pathogenic elements, and when we get rid of the pathogenic elements, we have a free seat to get hold of, and with the help of that particular seat, we can just like a cab, we can book it, and left it for the destination to arrive. So here in the case, we can just from the virus, we can make a therapeutic gene just by removing the non-required part, and hence we have the ideal system of vector, and we culture it and we package it, and then the vector virus is ready to be utilized. There are certain examples where we have used this approach, like for example, severe combined immunodeficiency virus, adenosine deaminase, cystic fibrosis we have used, familial hypercholesterolemia में भी हमने यूज़ किया है, thalassemia में we have used, sickle cell anemia में we have used, laxic, all these diseases you can get easily gene therapy से जो ठीक हुए हैं. So hopefully this helped you, and help you to learn better about gene therapy, and maybe it will help you in your questions as well. For more such mind maps, please subscribe the channel, and I'll see you in the next one. Thank you very much.